Thank you very much, Dr. Ahmed. Thank you very much, Dr. Ahmed. I'd like to thank you very much for your efforts, for your organization, for organizing this scientific program and your communication with the other colleagues to collaborate with in the successful scientific program. Today we will start the first lecture in the comprehensive neuroanatomy educational program, module number 4 for the spine and spinal cord. It's a weekly webinar organized by the Yemeni Neurosurgical Society in collaboration with the WFNS Young Neurosurgeons Forum and the Pakistani Society of Neurosurgeons, the Neurosurgery Couch, and the Neurosurgical TV. Today is the first lecture. It will be introduced by Dr. Nargiza Saatova. She's a neurosurgery forward Turkey, Istanbul, US, Florida, at Orlando Health Hospital. She's the founder of the Russian Neurosurgery Ground Round, and she will talk about the embryological development of the spine and spinal cord. Would you like to start, Dr. Nargiza? Are you ready to start?
Dr. Majid, hello. Thank you very much for the kind introduction. Yes, I'm going to share my screen.
We're glad to start.
All right. So can you see my screen?
Yes, it's clear.
Okay, yes, I'm going to start 1 second. So it's a great honor to give a first talk on the fourth model about the spinal and spinal cord pathology. So today I would like to talk about the embryology of spine and spinal cord. Just 1 second. So the presentation plan will be embryology of spine and spinal cord, and we will talk a little bit about the classification of congenital abnormalities. So these are the literature I was using while presenting and while preparing the presentation. So the development of body's form begins during the gastrulation, a process in which a trilaminar embryonic disc is created from the bilaminar disc occur during the 3rd week of gestation. It is well known that the notochord somites are the most significant structures responsible for the development of the future vertebral column. Epiplastic cells migrate from the deep surface of the primitive strix and form the embryonic endoderm. Subsequently, cells continue to migrate from a primitive strix, creating the embryonic mesoderm, and the cells that remain on the epiplastic side of the embryonic disc form the embryonic ectoderm. Cell migration continues into the 4th week of development, after which the primitive strix regress and disappears in the sacrococcygeal region. A group of specialized cells that migrate through the primitive node, which is located at the cranial end of the primitive strix, gives rise to the precaudal plate and notochordal process. The cells migrating most anteriorly form the precaudal plate, whereas the ones migrating most posteriorly form the notochordal process. The notochordal process develops into the notochord and will be an early representation of the future vertebrae and bony skeleton. The remaining cells from the notochordal process align themselves to create the notochordal plate, which will subsequently fold to form the notochord containing the central canal. The notochordal plate folds into the notochord. After that, the mesoderm surrounding these structures differentiate into paraxial, intermediate, and lateral mesoderms. Further, the differentiation creates from 42 to 44 pairs of somites from the paraxial mesoderm by the end of the 5th week, and eventually help in forming the bones of the head, vertebrae, other bony structures of the thorax, and associated musculature. They will be later on, all of them connected with vertebra and the vertebral processes. Each somit develops into two pairs, a sclerotome and a dermatome. The cells of the sclerotome are responsible for the formation of the spine, and the dermatomyotomes form muscle cells and the overlining dermis of the skin. The nerve system of the embryo is derived from the surface ectoderm of the trileminoid disc and begins to develop during the third week. Here you can see the picture of neural plate grooving, let's say, becoming the neural groove and then folding and eventually creating the neural tube. The several genes and the specific molecular signals from the notochord and mesoderm are capable of inducing the formation of the neural groove. The neural groove will fold into the tube and represent the future central nervous system. The brain develops from the most cranial aspect of the neural tube, and the spinal cord develops from the remaining caudal areas. Once the sclerotomes of that surrounded the notochord and neural tube, each level will separate into the cranial area of the loosely packed cells and the caudal area of density packed cells. Intervertebral disc will form between these two layers. You can emphasize it in the last fourth picture. Pia matter, about the matters, pia matter, arachnoid matter, and dura matter surround and protect the spinal cord. The pia and arachnoid matter are both neural crest in the origin, whereas the dura matter is the mesenchymal in their origin. In an embryo, the spinal cord fills the canal, but as a result of normal growth and development, it will eventually change the position. Now let's talk about the development of the spinal cord. The spinal cord develops from the neural tube caudal to the fourth pair of the somites. And here we have the Eller, means sensory plate. The Eller plate is a dorsolateral thickening of the intermediate zone of the neural tube. The Eller plate gives rise to the sensory neuroblast of the dorsal horn, general somatic efferent, and the general visceral efferent cell regions. So the Eller plate receives axons from the dorsal root ganglia that become the dorsal sensory roots. The Eller plate becomes the dorsal horn of the spinal cord. Then we have the basal or the motor plate. So the basal plate is the ventrolateral thickening of the intermediate zone of the neural tube. The basal plate gives the rise to motor neuroblast of the ventral and lateral horns, general somatic efferent, and general visceral efferent cell regions. The basal plate projects axons from the motor neuroblast, which exit the spinal cord and become the ventral motor roots. The basal plate becomes the ventral horn of the spinal cord. Then we have the sulcus limitans. The sulcus limitans is a longitudinal groove in the lateral wall of the neural tube that appears during the week 4 of development and separates the Eller and basal plates. The sulcus limitans disappears in an adult spinal cord, but is retained in the rhomboid fossa of the brainstem. The sulcus limitans extends from the spinal cord to the rostrum midbrain. Also, we have the roof plate. The roof plate is the non-neural roof of the central canal, which connects the two alar plates. The floor plate is the non-neural floor of the central canal, which connects the two basal plates. The floor plate contains the ventral white commissure. And here you can see the development of the spinal cord in 5th week, 8th week, and 10th week. Myelination of the spinal cord begins during the month 4 in the ventral roots, motor roots. Oligodendrocytes accomplish myelination in the central nervous system, and Schwann cell accomplish myelination in the peripheral nervous system. Myelination of the corticospinal tract is not completed until the end of the 2 years, when the corticospinal tracts become myelinated and functional. Myelination of the association neocortex extends to 30 years of age. Positional changes of the spinal cord. At week 8 of the development, the spinal cord extends the length of the ventral canal. At birth, the conus medullaris extends to the level of the third lumbar vertebrae. In adults, the conus medullaris terminates at L1, L2 interspace. Disparate growth between the vertebral column and the spinal cord results in the formation of the cauda equina, consisting the dorsal and ventral roots, which descend below the level of the conus medullaris. Disparate growth results in the non-neural filum terminale, which anchors the spinal cord to the coccyx. So the shape of the spinal cord will change the more fetus will grow, and later on. Classification of abnormalities. The neural tube defect failure of segmentation and formation. Spina bifida occulta, cystica, spina bifida meningocelli, spina bifida myelomeningocelli, myeloschisis, congenital scoliosis, congenital kyphosis, congenital lordosis, Klippel-Feil syndrome, congenital spondylolisthesis. A neural tube defect refers to a condition in which the neural tube fails to completely close during the 4th week of embryonic development. As a result, structures overlying these midline abnormalities are severely affected and may be unable to form. Failure of formation is the failure arises as a result of absence of a structural element of vertebrae. Any region of the vertebral ring may be affected. It can be anterior, anterolateral, posterior, posterolateral, and lateral. The type of deformity depends on the area of the vertebral ring affected, which will later alter the normal growth pattern. Typical observable defects are hemivertebrae or wedge vertebrae. Here you can see the pictures. Failure of the segmentation occur when 2 or more vertebrae fail to fully separate and divide the concomitant partial or complete loss of the growth plate. Embryologically, if 2 adjacent somites or their associate mesenchyme do not separate properly, a segmentation defect will occur. Spina bifida occulta is the result from the failure of fusion or development of part of the vertebral arch, usually lamina, which does not involve the spinal cord or meninges. Spina bifida with meningocelli, it's quite rare pathology, but still can occur. Patients developing spina bifida with meningocelli usually have a layer of normal epidermis covering the meninges. This lesion does not involve the spinal cord, and therefore these patients usually do not present with any neurological symptoms. External manifestation that may indicate this type of neural tube defect include their growth in the area of the lesion, lipomas and hemangiomas. Spina bifida with myelomeningocelli is more common than spina bifida with meningocelli. It is considered as a more neural tube defect because of the involvement of the spinal cord and meninges. This lesion may be covered by a thin layer of skin or by a membranous sac. Patients with this lesion exhibit typical neurological symptoms such as limb paralysis, as well as the bladder and bowel incontinence, and may also present with hip dislocations. Because this defect may involve just the nerve root of the entire spinal cord, paralysis may be of the flaccid, spastic, and mixed type. And other associated abnormalities may include hydrocephalus, and nuclear malformation, and severe forms of scoliosis, kyphosis, and lordosis. Myeloschisis is the more severe type of spina bifida. Myeloschisis is considered to be the most severe type of spina bifida. This defect occurs as a result of neural tube not closing properly with the development of the spinal cord being exposed to the external environment. Small patches of hair grow sometimes nails around the defect. Sorry, it's just small patches of the hair grow around the defect. As with spina bifida with myelomeningocele involved of the spinal cord translate into the possibility of one or more of the before mentioned associated defects, in addition to severe infection. Congenital scoliosis is an abnormal vertebral development that results in a lateral curvature of the spine is classified as congenital scoliosis. This type of spinal deformity occurs through the failure of formation or a failure of segmentation or the combination of both of them. Moore attributes the failure of formation to an embryological absence of one or more of the primary chondrification centers as a result of failure of cleavage of a primary center part of the vertebrae or growth plate will be unable to form the subsequent normal growth of contralateral side will create the lateral curvature. So you can see here the different types of failures of segmentation and formation, which eventually result in the scoliosis. Congenital kyphosis. Congenital kyphosis refers to a deformity in the sagittal plane, resulting in an excessive flexion of the affected area. Defects of this kind are classified as a failure of the formation, segmentation, or dislocation of the spine as a result of rotation. And as discussed previously, kyphosis with an associated scoliosis may be as well observed. The cause of this defect is also considered multifactorial in nature. So these are two types of kyphosis as a result of the formation at the first, and the second is the result of the failure of segmentation. Congenital lordosis, this defect is very uncommon and the least severe of the three abnormal curvature discussed in this review. The effect of this kind of creature abnormal extension of the spine. The only embryological defect associated with the true congenital lordosis is a defect of segmentation posteriorly. The cause of this defect multifactorial in nature. The most important syndrome, I would say, is Klippel-Feil syndrome. It is also called as a brevicolis, involved the congenital fusion of the two or more cervical vertebrae. Murray Klippel and his resident Henri Feil were the first orthopedicians who described this syndrome. This abnormality is a result of the failure of proper segmentation of vertebrae in the cervical region during the embryonic development. So here is the problem in segmentation. In addition, theories suggest that the defect is in the notochord, and notochord signaling may also cause this syndrome. Patients presenting with this defect have congenitally fused cervical vertebrae, causing the shortening of the neck with a low posterior neckline and diminished ability to move in the affected area. Patients are restricted to flexion and extension between the occiput and the atlas. Congenital spondylolisthesis. Spondylolisthesis is a condition that involves a forward slip of a vertebra and vertebrae in relation to the rest of the spinal column. Typically, this defect occurs between the fifth lumbar vertebrae and the sacrum. However, the spondylolisthesis has also been seen between the fourth and fifth lumbar vertebrae levels and may even occur in the cervical region or the higher lumbar levels. Studies showing an increase among family members, which suggest it as a genetic component. Thank you.
Thank you very much, Dr. Nargiza, for this presentation.
It
Dr.
won't
Majid?
be as a 40 or 60 minutes because the information is short. So today we decided not to discuss the clinical aspects, but embryological and what is the failure of embryological development can cause. I decided to put as a spina bifida and segmentation formation.
Thank you.
Thank you
Very
very much,
good
Dr.
lecture.
Nargiza. Actually,
Thank
So
you.
you was talking about a very important issue, embryological development of the neural system and its associated bony structures, which is the mainstay to understand the anatomy and of the central as well as the peripheral nervous system with the surrounding bony and anatomy and its intervertebral disc. As well as the development, the understanding the congenital anomalies which are related to the embryology, which is a very important issue, and we thank you very much to take the first lecture in this webinar, and we hope to continue the scientific program, inshallah, to the end with great success with the aid of the Dr. Ahmed Al-Jarad, inshallah. He's doing
Thank you so much.
very good job. Thank you.
Thank you very
Thank you
much.
so much.
Thank you very much, Professor
Thank you,
Amir.
Dr. Ahmed.
I appreciate these words. And thank you, Dr. Nargiza. Very nice presentation. About the clinical aspects of spinal dysraphism, it will be discussed in other topic in the schedule, inshallah, later. So if anyone have any questions or any remarks for Dr. Nargiza, please, you can open your mic and ask, or you can type in the chat box and I will ask.
Actually, Dr. Ahmed, I have just one comment regarding to the derivatives of the neural structures. The neural canal is the main stay in the development of the central nervous system, and it's covering the pia and arachnoid. While the neural crest is the mainstay in the development of the peripheral nervous system, most of the peripheral nervous system, except the motor neuron, which is derived from the central canal, from the anterior horn cell. While the somas is the responsible of the development of the bone structures, as well as the musculoskeletal system of the body, while the intervertebral disc is derived from the notochord itself.
Thank you so much.
Just to clarify this point. Thank you very much.
Thank you.
Thank you for the remark. If anyone have any question, please you can ask in the chat. See here. Someone, Dr. Muhammad Al-Sayri asked about what about DS2 material, split coat syndrome. I think he is asking about it if he didn't mention in the presentation.
No, I didn't mention that in the presentation. So I was deciding not to put too many pathologies. So
because in spinal dysraphism we have a lot of other diseases and other conditions like
split cord syndrome is not related to the failure of the segmentation and the formation. So it is the attachment. So it has different
mechanisms.
pathophysiology.
I think the statomyelia or the diplomyelia is related to the folding of the neural canal during its folding, as well as during migration of the cells from the somas, from the ventromedial aspect of the somas, while they're migrating to the front to form the vertebral body. This incomplete folding of the formation of the neural tube aberration during this process is the mainstay in the development of the diplo or diastatomyelia. If it's related just to the cord, it's diplomyelia, while if it's related to the migration of the somas, it develops the diastatomyelia, in which there is a bony septum in the spinal cord, dividing the cord into two separate parts with separate neural coverings.
We have also a question from Dr. Ashir Ali. He has asked about why are lipomyelinogenesis almost exclusively lumbosacral, and what embryological step fails in their formation?
I think I will be a little bit. I am in trouble to answer just because why it is only in lumbar region with Dr. Majid.
I think because of the process of the closure of the neural tube, actually it started at the cervical region and progressed caudally to the lumbar region. Most of the times of the neural tube defects are developed at the lower region of the spinal cord or at the lower region of the spine, because it's the end process. If there is incomplete closure of the tube, it will happen at the lumbosacral region. While if there is any failure during the process of closure, it can occur at any part, but mostly or most commonly it develops at the lower part of the neural tube, at the lumbosacral region, either spina bifida or the lipomyeloneuromeningocele, or in any other types. Even the spina bifida occulta or the aperta, they are developed almost at the lower part of the neural tube, at the lumbosacral region.
Thank you.
Thank you, Dr. Majid. Don't see here any further questions. So today's session will be short, I think, as first lecture, and it was very nice introduction to our program. So thank you very much, Dr. Nargiza, and thank you very much for your wonderful remarks, Dr. Majid Amer, Professor Majid Amer. Professor Majid is the president of the Yemeni Neurosurgical Society, and he's one of our mentors. Thank you very much, Doctor.
Appreciating your presence, Dr. Nargiza and Dr. Ahmed.
Thank you so much. I learned more from you today than I probably could present. Thank you so much.
Thanks for you. I hope you share with us in the future for more lectures regarding to the neural tissue, inshallah. Thank you very much, Dr. Nargiza.
Thank you so much.
Thank you, everyone. So I think by this we can conclude our first webinar of the comprehensive neuroanatomy educational course, and see you all, inshallah, next week. And we will have weekly presentations from now until 7th of August, inshallah. We'll have about 31 topics to be discussed. So see you all, inshallah, and thank you for attending today, and thank you for Dr. Majid Amer and for Dr. Nargiza for participating with us today.
Thank you so much. Thank you so much. See you next week. Thank you, Doctor. Bye-bye.